we will see that what are the criteria or parameters which are required for the secondary metabolite production. So please keep it in your mind. First, you are going to produce the biomass, that is your culture cells, and then you're going to produce your secondary metabolites. Secondary metabolites are produced in the stationary phase or in the EDO phase during the stress condition. When the microbial cells or plant cells are under the stress condition, then the secondary metabolites are produced. These secondary metabolites are important for the plant only for the defense mechanism. They are not necessary for the growth and development of the plant. Okay, and these secondary metabolites has various application, as you saw in the previous class only, in the pharmaceuticals, in the as a food additives, flavors, enhancers, antimicrobial agents, or many things like antifungal, antioxidant. Okay, so in order to produce the secondary metabolite, what are the parameters which are important so that we can produce the secondary metabolites in high amount efficiently? So the second stage is the secondary metabolite production. For this, we need to change the culture medium. We have to make it more rate limiting, or in other words, we are inhibiting the cell growth by reducing the phosphate level, nitrogen level, or we can add various adjuvants to the culture. So what I'm doing is I'm going to change the media. The media which I used for biomass production, I cannot use it for secondary metabolites. I want to use a rate limiting media which is going to reduce the nutrients. Once the nutrients are reduced, what happens? The cells are under stress. They are not able to get sufficient amount of nutrients. They are under the stress condition. How I can bring about the stress condition is by reducing your phosphate level, nitrogen level, or we can add various adjuvants to the cells which can bring about the stress. So first we'll discuss about the precursor feeding. Here we are making the environment for the cells more challenging. That are, we are making the cells grow and in a condition which can inhibit their growth. So what we're doing, we are creating a stress which forces the cells to start producing the secondary metabolites. So this is the first step in the precursor feeding. In precursor feeding, we know that in biosynthesis pathway, because all the cells will be having their metabolic pathway. So in the biosynthetic pathway, one biomolecule serves as a precursor for the next reaction. So you have series of your metabolic reaction, and you have seen that a substrate of one is converted into the product of another by the enzymatic reaction. So one of the substrate is acting as a precursor for the formation of the next reaction. So depending on the metabolic pathway of the secondary metabolites produced by the cell, precursors are added in the media, which are then converted to the desired product. How they are converted to the desired product means they are converted by the pre-existing enzymes in the cells. So the enzymes are already present in the cells. When you're adding precursors, what will happen? The enzymes will take up the precursors and they will convert it, because you know what type of precursors you are adding, because you want the secondary metabolites of what you have decided, right? So you are adding a precursors to get your decided products. So already the enzymes are pre-existing in the cells. They will use the precursors and they will convert those precursors into biomolecules which you require. That is your decided product you be getting. So these precursors are very important and critical. For example, they have done a study in a plant known as Catranthus roseus. They have showed that the increase in the secondary metabolite production by addition of certain precursors. So when they added certain precursors, this Catranthus roseus, what it did was it produced more secondary metabolites. So this is one of the example of the precursor feeding. Next is the elicitors. So Why are these elicitors required? The second parameter what we required is the elicitors. Now, what we understand by elicitors means they stimulate the biomolecules. That is, they are going to stimulate your secondary metabolites. And the process wherein the elicitors stimulates the secondary metabolite production, they are known, the process is known as elicitation. You can categorize these elicitors into the biotic or abiotic elicitors. For example, fungal or your bacterial origin can act as a biotic elicitors, whereas your abiotic elicitors can be like inorganic salts, heavy metals, some UV radiation, high salinity, high loss osmolality, extreme temperature, high pressure. So there are multiple ways by which you know to trigger the cells. So what we are doing, we are triggering the cells in such a way that we are putting them into stress so that they are able to produce the secondary metabolites. So elicitor stimulates the secondary metabolite production by addition of abiotic or biotic elicitors. So these are all bringing stress to your cells so that the cells can produce the secondary metabolites. So the cell dividing actually stops and they start producing the secondary metabolites in the stationary phase. And so what happens is when you are adding the elicitors, the cells release the products into the medium. So elicitors are mostly applied during the growth phase, that is during the exponential phase, elicitors are added. Okay, because that time only you have to add, because during the exponential phase or in the log phase only your cells are dividing. So I want my cells to divide less and enter into the secondary phase to produce the secondary metabolites. That is my strategy what I am going to use. Next, another method is the biotransformation. Now, what is this biotransformation? Biotransformation, now it is a process by which an exogenously added compound is transformed to a high value product by the culture. So you should know that plant cells or your plant cell cultures and enzymes have the potential to transform inexpensive and plentiful substances such as industrial products into rare and expensive products. Okay, this you have to remember. So what happens is there are many products in the plant. Okay, plentiful are present. So what you do is you have to convert them, you have to transform them in this all these plentiful substances into industrial product which should be rare, and these are quite expensive product. So what these products are already present but they are not what showing up, so you have to add something so that they will produce such important compounds. So what we do is we use the biotransformation. It is a process by which exogenously added compound is transformed to a high value product by the culture cells. So say there are certain products in my plant already, but they are very inexpensive and they're not producing any valuable compound. So what you do, you add certain compounds exogenously, you add by yourself into the media. Then what it will do, that product whatever you have added, they're going to interact with your plant products which are inexpensive products, and they will convert them into a highly valuable product and which are expensive, so that it can be industrially it can be utilized. One example what they have told is the Arbutin. See, the spelling has gone wrong here. It is AR, there is no D. Okay. So, arbutin is a secondary metabolite which is produced again by a in the plant known as Catranthus roseus. So, what they have done is the scientists have added hydroquinone into the cell cultures of the plant, catharanthus. So when they added, what they found was it was able, that cell knows they were able to produce this arbutin, and this arbutin has having very important economical properties because it is extensively used in the cosmetic creams and it is used in the skin lightening agent. Okay, so this is one of the example for how you're bringing about biotransformation. That is, you add your compound exogenously and then try to convert the inexpensive compound which is already present in your plant or which are present in very small quantity in the plant into a high expensive products. Okay, one example is the arbutin which is produced in the catharanthus by adding hydroquinone. So the next one, what we are going to see is that immobilization of cells. We know that in vitro cell suspension cultures can be used for commercialization of secondary metabolite based product. But the problem with your suspension culture is all the cells are unsynchronized cells. They are not synchronized. That is, they are not in the same stage of cell division. That is called as unsynchronized cell division, okay, or unsynchronized cell growth. Not only that, another drawback of your suspension culture is the cells are non-viable and the product yield is very low and they are genetically unstable, and the selected line show the cell lines show low shear resistance of the cell, and whatever the metabolites are produced, they are produced internally. Okay, intercellular metabolites are what they are produced. That is, the cells produce the secondary metabolites within their body itself. So these are the some of the drawbacks when we do it in the suspension culture. So what we have to do is we have to use a technique wherein these drawbacks are inhibited. We remove these drawbacks. Therefore, we use this technique called as immobilization, which is having a lot of advantages. Like it helps in the extending the viability of the cells in the stationary phase. We told that the cells will not be viable, but by using immobilization, what we are doing is we are seeing to that the viability of the cells in the stationary phase, and thus enables the maintenance of the biomass for prolonged time period. It simplifies the downstream processing, and in the case where the products are secreted in the medium. So when the products, you see to that instead of product being secreted within the cells, if it is secreted in the media, it will be easy for the downstream processing. What are the downstream processing? Downstream processing means a product is formed and you have to make that product more purified because it is present, a product which is formed is present along with the media and other toxic substance. I don't want all of them, I want pure product, and the purification process is known as the downstream processing. And if the high density, if your cells are in high density, you can use relatively small reactors, okay, which results in the reduced cost and risk of contamination. It reduces the shear stress. We told that suspension culture causes shear stress. Okay, shear stress. What is the shear stress means? It is a force tending to cause deformation of your material. So there is a force which is causing your material to become deformated. That should not happen, deformation. My material should be stable and it should be in the original form. It should not change into deformation stage. That is called as the shear stress. So it reduces the shear stress. Which reduces the shear stress? Your immobilized enzymes when you're using it in small reactors. Okay, it increases the product accumulation and it minimizes the fluid viscosity. If there is fluid viscosity, what happens? It causes the mixing and aeration, a problem in cell suspension. So now it helps in increasing the cell density by 2 to 4 times more, and it separates the cells from the media. And so it is very much relevant in the process of biphasic. What is biphasic? Biphasic is such type of whatever we are explaining. That is, first you're producing your biomass and then you're producing your secondary metabolites. That process is called as biphasing. So you can see the advantages of using immobilized cells. So these are all this what I spoke, and here also what I spoke is all the what positivity, that is the these are very much important for the production of the secondary metabolites. So I can use the immobilized enzyme and reduce all the negative aspects in the reactors or in the cells. So if these are all the function of the immobilized enzyme, they're bringing such a good product formation. How can you immobilize your cells? So immobilization is of two type. One is the gel entrapment method and the second is the surface immobilization method. Most commonly what they use is the gel entrapment method. Okay. So what you do is in gel entrapment method, herein, the cells are entrapped in a specific gel or in the combination of gel, and then it is allowed to polymerize. And the gels which are used for the entrapment are the alginates, agar, agarose, carganin. All these are used as a entrapment. These are all gels. Okay. So what happens is they polymerize, okay, and they make form such lattice. When they form such lattice, what happens, um, your cell cultures, they get entrapped. That is, they are called as immobilized. Now the cells cannot move. Okay, so this is one of the methods wherein you can immobilize the cells. So by immobilizing, what you're getting all this positive effects. Okay, so another method is the surface immobilization method. This takes advantages of the culture cells. Okay, that is surface immobilization. You have a particular surface. For example, you can have the surface as polyurethane, foam fiberglass, mats, and woven short fiber polyesters. These are all what? These are surface immobilizers. So what happens is your cell culture, no, they can adhere to these surfaces, all the surfaces, and your surfaces will not have any adverse effect on your culture. They are inert. Only what happens is your cell cultures go and attach to the surfaces. Now, once they go and attach to the surfaces of these, whatever I told, the fibers under, what happens? They are become immobilized. They cannot move. Now the cells are trapped. So once the cells are trapped, what I can do is I can bring about all this variation in the um, production of the secondary metabolites. So the next parameter what is necessary for your secondary metabolite is the permeabilization. Permeabilization of the cell, we know that the plant secondary metabolites are largely produced intracellularly, and extraction of such products into the medium is very important because the removal of secondary metabolites continuously from the media increases the productivity. It allows the repeated use of culture. So once the product is produced inside the cell, you see that the products are not produced in the cells, but they are excreted into your media. So that only your downstream processes can be simplified. Okay, and when you're doing when you keep on removing the your metabolites, what happens? It increases the productivity. Okay, and it allows to use your culture many times. Okay, so you have different agents are used for permeabilization. And what are they? They are like DMSO, reducing the pH, sonication, and electric shock. Okay, so when you go for sonication or electric shock, what happens? The cell starts to break, the cells will disrupt. Once the cells disrupt, what happened? It will release whatever contents which was inside the cell into the media. So my secondary metabolites also come into the media. Now I can extract it by the process of purification, which is known as downstream processing. And the another parameter what we use is the selective absorption. Hope this is understood. I'll just draw the pictorial diagram. See, this is your cell, no? Your cell is producing all the secondary metabolites. Along with your secondary metabolites, there will be the own composition of the cell itself. Now, suppose say I'm using this DMSO, pH, reducing the pH, sonication, electric shock when I give to the cells, what happen? They will get disrupted. Okay, they are damaged. Once they're damaged, what happen? All this is released. Where they'll be released? They'll be released into the media because they're growing in the media. From the media, you can extract the process by the downstream method. The next method is the selective adsorption of the metabolites. So selective adsorption of metabolite has been observed that the secondary metabolites which are produced in the cell culture should be removed quickly. Otherwise, it will lead to feedback inhibition, enzymatic or non-enzymatic degradation of the product in the media, or volatility of the compounds produced. So it is very important to develop a separation technique that can concentrate your decided product. So some of the products which are used for removal and to concentrate your secondary metabolites are, um, see here it is like polycarboxylic esters, resins, neutral polymeric resins, activated charcoal, polyethylene glycol, and cyclodextrin is used. So I'll explain again. This is another parameter called as selective absorption of metabolites. So what is happening when the metabolites are being produced, secondary metabolites are produced in your media or in the in vitro culture, what you have to do is immediately you have to start to remove the secondary metabolites. You have to remove. If you don't remove your secondary metabolites, what happens? Okay, it can lead to the feedback inhibition. That is, whatever the products are being formed, the toxics, it becomes like a toxic and they'll start inhibiting your product itself. Okay, so a product formed can inhibit the substrate of another one. So I'll just see if this is your substrate and this is your product. Again, this product will act as a substrate for another product formation. So what is happening? You're not removing your product. That is your secondary metabolism. Now what will happen? This product itself can go and attack your substrate. That is called as a feedback inhibition. And also the cells are having enzyme and non-enzymatic materials. They also can go and inhibit your secondary metabolites. That is why you have to, as quickly as possible, you have to remove your secondary metabolites. And these all materials what I have mentioned here, sorry, this all materials like charcoal, polyethylene glycol, cyclodextrins, these are all used for selective adsorption of your metabolites so that your metabolites will get adsorbed to these materials and you can easily extract your secondary metabolites. So the next technology what we are going to speak about is the hairy root culture. Okay, see, this is the hairy root culture. It is a tissue culture method. Hairy root. So this hairy root culture, this is because we know that why we are using this hairy root culture is this is because we know that the secondary metabolites produced in cell suspension culture has number of limitation. Okay, such as the cell population is not stable, then there is they undergo mutation and the yields also are low. When there occurs mutation, what happens? The yields get affected. Also we know that the secondary metabolite production is localized. That is, they are produced only in certain organs in some plant. So, hairy root culture is another technique to avoid all the limitation what you are observing in the cell suspension culture. So, remember, students, you can produce a cell secondary metabolites either using suspension culture. That is my first video what I did. Otherwise, because the suspension cultures have lot of drawbacks, you can use immobilized enzyme to remove all the drawbacks. But still, another better technique is to use the hairy root culture. Okay, it will try to avoid whatever problem is there in the suspension culture. The hairy root process is an automated process, and we can achieve high mass of roots using transformation technique, in which we infect the tissue with a bacteria called as the agrobacterium rhizogenes. So what will we do is, we'll take any part of the tissue, that is your explant. Okay, this tissue will take, and we are going to infect it with the bacterium, and the bacteria's name is agrobacterium. Okay, rhizogenes. Rhizogenes. So what I'll do is, I will infect my tissue with the bacterium, and that bacterium is called as agrobacterium rhizogene. The very word is telling that this bacteria has affinity towards the roots. It can infect the roots. So under in vitro condition, we are going to infect the tissues with the rhizogenes. So you can see here, once if I infect my tissues with this agrobacterium, what happens? Your roots now start to branch. You can see that branching has gone extensively branching. They are genetically stable, and their growth rate is very high. So whenever I'm producing the hairy root culture by using agrobacterium rhizogenes, I will get the roots which are extensively branched, and they are genetically stable, and the growth rate is also very high. So now for this method, we are going to take a plant. How we are going to produce this hairy root means, we are going to take a plant whose genotype is already analyzed. Okay, you know that how we are going to analyze by chemical profile. We are going to analyze the genotype of the plant. So the genotype is already analyzed, and it should have a high secondary metabolite production. So we take that tissue. So suppose say this is the plant, and this plant is having a very good production of secondary metabolites. Okay, so what we are going to take, we are going to take this plant's explant. Okay, explant means, you know, it is a part of the, it is explant, ex. Okay, explant, it may be any part of the tissue of the plant. And then what I am going to do is, I'm going to sterilize this explant, and then after sterilizing the explant, I shall write it and show you all. So I've taken the explant. After this, I have to sterilize the explant. After I sterilize the explant, what I'm going to do, I'm going to create a wound, wound, where I'm going to create the wound in the explant. And where did I get the explant? I got it from the plant, which is having a genotype which can produce a high secondary metabolites. Okay, I have formed the wound. Then what I'm going to do is, I am going to co-culture it with the agrobacterium rhizogenes, which in turn induces this type of adventitious root from a tissue. So from a tissue, I am getting a adventitious root. It does not mean that you have to you have to have only root initial. So it can induce root in any kind of tissues, and we can incubate the infected tissue in the idle condition. So the tissue starts producing adventitious root. In the next stage, we will excise, we will remove, okay, that part of the adventitious root, and we will culture it in the next media. which is would be to stimulate more roots. So I will cut this root. Okay, so now I have cut all these roots. I have this egg-sized root. Now what I'm going to do is I'm going to grow it in a culture media. This culture media is different from my previous culture media. Okay, what is this culture media, how it is different? Now this culture media is going to produce more roots. So once you have more roots, okay, many more roots are going to produce. All Many more roots are produced. So where are these roots all produced? Either in your conical flask, or you can grow it even in the petri plates. So we are going to take it to the next stage. Now so many roots are produced here. Now I have to take it to the next stage. The next stage is known as the bioreactors. You know that bioreactors are vessels. Okay, such type of vessels. Okay, it is called as an it is fully controlled. It is just like a factory to you're generating. For example, say you there is a factory like what we say Suzuki, uh, which is going to produce two wheelers or four wheelers. Like that, here it is a factory which is going to produce only your cells. Okay, these bioreactors. So this bioreactors is an automated system wherein all the parameters are controlled. The nutrients, the culture condition, the physical parameters are all controlled so that there is an optimal production of the product of interest. So our biomass and secondary metabolites are now produced. Okay, because you have to produce biomass, that is your root culture. Okay, so once I produce my root culture, I saw to that I produce more root culture by changing the media so that I'll get more number of adventitious root. After I got the adventitious root, I put it into the bioreactor, which is a automatically controlled system. Here what I saw to that, more biomass is produced, and simultaneously I'll change the media such that it can produce what, your secondary metabolites. Okay, so a biomass and secondary metabolites are now produced in the bioreactor. That is, now we are going to change the media to the rate limiting media. Why rate limiting media? So I told you carbon, nitrogen, phosphorus, these are very much required for your microorganisms or for cell. And if you're going to limit that nutrient, what is going to happen? The nutrient will not be sufficient. The cells will enter into the stage of stationary phase. That is, they are moving into the death phase. And I told you stationary phase is what we want because that is where the secondary metabolites are going to produce. So I'm going to transfer the media, change the media so that I will limit the nutrients. Once I limit the nutrients, my cells are undergoing stress or traumatic shock, they are undergoing, and they start producing the secondary metabolites as a source of defense. So once the hairy root is produced, we transfer it to the secondary bioreactor, which the conditions are made this rate limiting. Okay, so that the root culture starts producing the secondary metabolites. Thereafter, the entire roots are harvested and used as for downstream processing. So the production in the bioreactor is relatively high. Okay, whatever products you're getting, no, it is very high and very pure. It is very many fold high than what you get it in the other methods like suspension culture or any other methods. But one problem with the bioreactor is it is very costly. But when you compare that is the cost of investment is very high. Okay, but what happens is, suppose say I am investing my money in the bioreactors, it will be very costly for me. But the end product, what I'm going to get, no, that secondary metabolite, it is highly expensive secondary metabolites. See, whatever I told you, pharmaceutical, in the food industry, or in any other industry, we use this all secondary metabolites only. Okay, your drugs, your antibiotics, your what is that flavors, and resins, waxes, all these are secondary metabolites. Are they not industrially important, commercially important? Yes, we all are buying that. And so when people are buying from me, what happens? Whatever investment I've done, I'm getting it back. Okay, so now what the scientists have done is one more method is they use two bioreactors. In one bioreactor, you're going to produce only the biomass, and in second bioreactor, you're going to produce only the secondary metabolites. And here, what this picture is telling you is this is the lab scale they produce the root. Okay, that is establishment and optimization of the study is being conducted in the shade flask. First, you see that everything you produce in small quantities in your shade flask. That is the research scale. Okay. Then you infect your roots or the explants with that of the agrobacterium. You sterilize your explant, produce a wound in it, and then infect with your agrobacterium rhizogens. This agrobacterium rhizogens, then what will happen? It will start multiplying in the explant. Once it multiplies, what happens? It's going to produce only roots. And what type of roots? It is the adventitious root, and it is highly branched. Next, what you do is you take this roots and you culture it in another media which can produce more number of roots. Now, when it is successful in the lab scale, only then you go for the a pilot scale, wherein you are going to use small scale fermenters like this. Okay, so you can use the culture media for growing the biomass, and then you can use change the culture media, that is you limit your nutrients such that it produces what? It produces the secondary metabolites. And when year and year, if my experiment is successful, then I can produce it in large liters, large quantities, 10 liters, okay, like 10 lakh liters, 20 lakh liters, I can produce the product in the final in industrial scale, because this is not sufficient, right? Whatever we are producing here, it is small quantity, so I want it in very large quantities. So I'll be using a very large fermenters like this. So students, like there are many types of fermenters, okay, which you can use. See, one of the drawbacks, what I told of using bioreactors is it is very costly. But when you compare to your metabolites which is produced, your cost is nothing in front of it. Because when you get very good secondary metabolites, you can sell that product to your consumer and you can get back your capital investment. Plus, you will get back the profit also because you are going to sell it in uh, high rate. Okay, and there are some problems even in the bioreactors. Okay, it is not that all bioreactors you'll get excellent results. Only it has to be standardized and it should be optimized. Some of the problem is that culture is greatly affected by the aggregation of the cells, which eventually results in the settling down. Now, if your cells are, see, your cells should be like this, separate, separate. But if they are aggregate like this, what happens? The density increases and they are going to settle here. When they are going to settle here, how can they act on your substrate and produce product? They cannot. So this is one of the drawback. And there is non-uniform availability of nutrients and oxygen, and eventually it can decline the secondary metabolite production. So there are many reactors also. Okay, each industry can use each type of bioreactors according to their profit. So there are different types of bioreactors which have their own pros and cons. Okay, such as most commonly used is the stir tank bioreactor. Otherwise, you can go for rotary drum bioreactor, bubble column reactor, trickle bed reactor, hairy root culture, gas phase bioreactors, spray droplet reactors, mist reactors, balloon type reactors. Now just imagine how many bioreactors are there, right? So why there are so many bioreactors means there are so many products you can produce, and you can't use only one bioreactors, okay, for producing all the metabolites. Okay, you have to change some of the parameters in your bioreactor so that you get the decided products. One simple example I'll tell you. See, you are cooking the food. Okay, you have cooked the food and in a vessel. Are you going to cook your curry also in the same vessel which is containing the food? No, you use a separate vessel, right? Because you know that I can cook my food in the rice cooker, okay? But other curries or gravies, what I want to do, I can prepare it in the another vessel. It is like that. Based on what is decided, you can use the type of bioreactors. So ultimately, ultimately is I want the product. What is my product? Secondary metabolites. So remember, students, before we wind up, see, look at this beautiful root culture, how much they have been produced, adventitious root, and imagine how much of secondary metabolites they'll be carrying. Okay, this one then I'll use it for the large scale bioreactors like this. So next time, whenever you are using anything, okay, any resins or vaccines, you have to remember that how much process is going into it when you're taking your tablets, okay, when you're adding your flavors for your uh, or enhancers, when you're using this preparing sweets and all. Look, all these are produced by your secondary metabolites process. So first you produce your biomass, second you produce your secondary metabolites. And I have taken the example of the plants. You can use even the microorganisms. So some of the commercially important metabolites are like taxon, chiconin, scopolamine, digoxin, piperine, morphine, quinine. Okay, all these are mostly drug components which are produced.